Thank you, everyone. Today we've got a very special speaker, so Professor Justin Stebing. Professor Stebing is really a multi-talented, multi-mind thinker who uses his gifts for many different areas, and his main focus is being a Professor of Oncology and consultant at Imperial College, and a translational professor also at Imperial College. And if it doesn't not busy enough, he's also editor-in-chief of Oncogene and senior advisor at Benevolent AI. And in January 2020, an early publication in The Lancet led to an FDA-approved treatment for COVID-19. So Professor Stebing has really made a huge impact upon the world in many different ways, both in cancer and in COVID, and he's still relatively young. So there'll be lots more to come, and our moderator and questioner afterwards is the eminent Dr. Plowman from St. Bartholomew's. I think most of the audience will have seen or heard of Dr. Plowman, esteemed in his own right. So Professor Stebing, over to you on CTCs. What tests can we perform on them, and what is their clinical use? Thank you for your time. Over to you.
Thanks so much, Nathan, and thanks to Angle for sponsoring this, and Nick, thanks for moderating this. So I'm going to talk about circulating tumor cells really from the very beginning, including, I'm really sorry to say, quite a few of my own publications in the area, just to help us think about them and see how we can frame things. Now, when it comes to circulating tumor cells, the basic simple thought is, what are they? We call them circulating tumor cells, which implies they're just cancer cells in the blood. Normal people shouldn't have them anywhere. Lots is said about them. The simple view was always that they're released from a primary tumor and that they're en route to forming a secondary metastatic focus. And there's many ways of classifying them. They form clusters, for example. And studying them really gives us an insight into the way cancers behave in transit, because primary cancers are typically curable, but when they spread, metastatic cancers are typically incurable. Now, CTCs can be used for all sorts of things. They can be used for screening and early detection of cancer. They can be used to estimate the risk of metastatic disease. We call that prognosis, how long someone's going to live for. But they can also be used for stratification and real-time monitoring of therapies and therapeutic benefit. And really, I'm thinking there about prediction, deciding if a treatment's going to work or not. They can help identify drugs to treat patients, guiding clinical trials, and provide biological insights into the genomics of tumors. And we know tumors are heterogeneous. They're not just one cell. They're many different types of cells. And in doing so, they help us understand the biology of metastatic development. Now, this is an editorial I wrote at the bottom in The Lancet in 2009, where I described them as more than prognostic markers. And there's an interesting quote I wanted to bring it to your attention. And it was a note on Albert Einstein's wall at Princeton, which I think is very apt for when it comes to talking about CTCs. And he said, or the note said rather, not everything that can be counted counts, and not everything that counts can be counted, which I think is really very, very profound indeed. Now, when you think about circulating tumor cells, I want you to think of them as often giving us more questions than answers, and often giving us answers. So at around the same time, we were looking for where they lived in the body. And what was interesting is when surgeons operated on hepatobiliary tumors, we were sampling the portal vein and the blood vessels around the liver and the pancreas, and we found really, really huge levels of circulating tumor cells, much greater than when we were sampling typical peripheral veins. And if you look on the left, the conclusion of this paper was that in patients with colorectal liver metastases, circulating tumor cells were localized to the hepatic circulation. And interestingly, That's maybe one reason why we never or rarely see patients with solid malignancies with metastases in their arms or legs. I published a paper showing that if we found these cells in people with indeterminate lesions of the pancreas, so sometimes on imaging we'll see pancreatic lesions and we don't know if it's chronic pancreatitis, a cyst, or a cancer, then it tells us that these are pancreatic cancers. But these are dynamic real-time events. So when surgeons don't manipulate tumors that much during their resections, you get fewer circulating tumor cells released. And these were all very interesting early insights that these are functional, active cellular moieties. And at Imperial College, we started cleverly designing trials around them, linking to the mechanism of action. So lapatinib, for example, is a drug licensed in breast cancer that inhibits both HER2, a very well-known breast cancer target, but also the epidermal growth factor receptor, the EGFR. And we wanted to see in metastatic breast cancer if it would work decreasing EGFR positive, epidermal growth factor receptor positive circulating tumor cells, as well as HER2 positive circulating tumor cells. And we started evaluating this drug in all sorts of patients. So these were patients, and we know tumors are heterogeneous, with HER2 negative tumors, but HER2 positive circulating tumor cells, with HER2 negative tumors, but EGFR positive circulating tumor cells and HER2 negative ones, and different combinations of them. But what we found was that although you could deplete the peripheral EGFR positive and HER2 positive circulating tumor cell pool, if the primary tumor didn't express those markers, it didn't affect the primary tumor. And mechanistically, this makes sense because you have the primary tumor spreading via a process called the epithelial mesenchymal transition, transitioning as it becomes more aggressive from an epithelial phenotype to a mesenchymal phenotype. And this is when we think circulating tumor cells are released. Now, why is it that primary tumors such as breast cancer or colon cancer or prostate cancer after surgical removal can relapse many years later? Well, there's two possibilities. Either one possibility is it's like compound interest, that a circulating tumor cell elsewhere or a disseminated tumor cell can exist in the periphery and years later start growing again. So one cell becomes 2, which becomes 4, 8, 16, 32, 64, 1024, so on and so forth. Or maybe the intriguing possibility is that a circulating tumor cell can behave like a cancer stem cell, and 10 years later, like a queen bee, can repopulate the entire nest. Now, measuring circulating tumor cells is an enormous technical challenge because when you take the blood of a patient, even with widespread cancer, you're essentially looking for one cell in 10 million. And the way you identify them is you're looking at the biological properties of the cells and the protein expression, and also the physical properties. And there's many different ways to measure them. But unfortunately, nearly all of the methods to measure them end up killing them. So you can't easily study them functionally. They come out dead. Now you can study them via positive selection, looking for markers on tumor cells, or negative selection. So for example, in the bottom left, you can see a stain for anti-CD45. Now that's a well-known white cell stain. And if that's present, we know it's not a circulating tumor cell, but a white cell, and they have similar sizes. Various people make organs on a chip or labs on a chip, and that can be used as well. And at the end of the talk, I'm going to talk about something very simple, but often the best things in life, as you know, are simple, which is just selection of circulating tumor cells on the basis of size without killing them. Now, there's lots of methods that have been tried. Immunocytochemistry has been tried, looking at markers such as vimentin and cadherin, tissue-specific markers such as mammoglobin, and of course tumor markers such as HER2. There's molecular technologies where individual sequencing can be performed on them, and if they have deranged genes such as mutations in RAS, the commonest oncogenes, or deletions in p53, the commonest tumor suppressor gene, that indicates that they're circulating tumor cells, and they can be extracted from the blood and occasionally cultured to see if they're functional. Now, the FDA has approved one test, which is the Veridex cell search system, and it's approved it in breast, prostate, and colon cancer just for prognosis. That's all. It's a very big machine. I've got one in my lab at the Hammersmith Hospital. It's expensive to use as well, and it purely provides a quantitative number, although now they make cell surface markers such as EGFR stains, HER2 stains, and others. And the biochemistry makes sense. So you're putting your tube of blood in, and immediately you're staining the blood for DAPI, which is a nuclear stain. And you may remember red cells are anucleate, not in birds, but in humans. So you exclude all the DAPI negative cells. You just want the nucleated cells. As you know, I'd mentioned that CD45 is a white cell stain, so you keep those. And then you're looking for CK positive cells as well, cytokeratin positive cells. And we believe these are tumor cells, and typical cytokeratins are cytokeratins 8, 18, and 19, for example. Now, as mentioned, the main strengths are prognostic data in breast, colon, and prostate cancer, which simply tells you how long people are going to live. And these are very big data sets. So one of the first ones, Christopher Nelly, published in the New England Journal of Medicine, simply showing that if you have less than 5 circulating tumor cells in a 7.5 mil EDTA type tube of blood, you're going to have a good prognosis. But if you have more than 5 circulating tumor cells, you'll have a poor prognosis. In colon cancer, it's harder to get circulating tumor cells, so the cutoff was drawn at 3. And at prostate cancer, Johan de Bono, who studied CTCs in a lot of clinical trials, has a cutoff at 5. Now, I did some work looking at the role of circulating tumor cells in dying people. These were people admitted to hospices and to the wards where no further treatment was being offered, and they were considered to have less than 6 months to live. And patients' relatives were asking me, how long has this person got, doc? And it was always said, well, you know, it could be days, weeks, maybe a few months, but it was very, very difficult to say anything. But what we found was that if there were more than 100 circulating tumor cells, the patient was about to die and had a median survival of 17 days. But if they had less than 100 circulating tumor cells, they had a median survival of 182 days. And this was across many different metastatic cancers in people with very heavy volume disease. So it became clinically useful just talking to patients' relatives. Now, this is data that I published in the Journal of the National Cancer Institute. And it's not as simple as more than 5 and less than 5. This was published last year, big, wide collaborations in metastatic breast cancer. And what we can see is each increase in the number of circulating tumor cells, so going from 5 to 6, 6 to 7, 7 to 8, 8 to 9, and so on, is associated with a worsening of prognosis. That's important. But what's especially important is they're also predictive. Now, what does that mean in this context? What this means is that if your treatment takes you from more to 5 to less than 5, it's like you started with less than 5 in the first place. So it's telling you if your treatment is working. So beforehand, I mentioned that you can tailor-make different clinical trials and different therapeutics around the surface markers of circulating tumor cells. But here what I'm saying is, not only are they prognostic, telling you how long people might live for, but they're predictive. It tells you how to treat people. So if your treatment didn't take someone from more than 5 to less than 5, then maybe you should switch treatments, and circulating tumor cells gives you a quicker guide, and they change before any radiologic changes occur. And this continues over time, so it doesn't matter when you're measuring them. It doesn't have to be at baseline. It can really be at any time. And this study here shows that rising circulating tumor cells at 3 weeks was associated with a worsening overall survival. And again, that's predictive because it tells you what to do. It tells you about future directions and optimization of therapy as appropriate. Now, these aren't just static cells. The future is their genetic characterization, not just their detection, not just their counting. But there's not that many platforms that you can do this on. Now, one of the problems I'd mentioned earlier was that many ways of collecting circulating tumor cells leads them to die in the processing. So it's very important to find ways where you can collect circulating tumor cells without killing them, so you can perform single cell genomics, study the genes, study the surface markers to really understand cancer better. And it's not just about understanding cancer better, it's about performing functional studies. So you can take circulating tumor cells that are HER2 positive and inject them into mice, and they spread to the brain. We know that circulating tumor cells, like other cells, release exosomes, little packets, little lipid particles containing DNA, RNA, and protein that prime metastatic sites, that prime secondary sites for them resting in and growing secondary tumors. But you can also take them out and perform studies of drug susceptibility and sensitivity, and you can look at their tumorigenicity and their aggressiveness by studying them. And you can do this across tumors, including tumors on the border of being epithelial tumors. So there's different kits, and you can study small cell lung cancer and melanoma, which are not necessarily considered epithelial tumors, and even sarcomas as well. You can even generate various lines from them. Now, new technologies are often the most simple. One technology I particularly like is Angor's Prostate technology, which is basically based on simple size requirements of circulating tumor cells. And you can harvest CTCs, stabilize them, but you can also study them because you're not killing them, which is a really, really crucial feature of these cells. So in conclusion, they're useful clinically, they're useful in research, they give us additional information. They're much more helpful than tumor markers such as CA15-3CA or LDH. They're fascinating biologically, they're really, really interesting and potentially very useful at directing treatment as well as for prognostic and predictive information. We have lots to learn about them. As this picture on the right shows, they also finally provide different opportunities for therapeutic intervention for drugs that specifically target CTC pools, because CTCs are probably the primary tumor's way of spreading to other places in the body. Thank you very much indeed. Nick, over to you.
Fascinating. Thank you very much, Justin. Justin, I think we're going to have some questions from the audience. I hope so. But can I start off? Do you think that the clinician needs next generation sequencing on tissue and on CTCs to find out what the primary tumor is doing and what the leading edge is doing?
I think that's a great question. And do you want to just clarify what you mean by the leading edge? I think I know what you mean, but do you want to just explain to the audience what you mean by that? Because I think that's very important.
Colon cancer, you can see budding of the peripheral edge of the tumors, budding cells, which then become intravascular. This is what I think is known as the leading edge, and it's thought to be, as you say, the place where epithelial to mesenchyme transition occurs, or even the reverse under therapeutic circumstances. And as you pointed out, that may be the determining factor of metastatic potential. So is the change in the phenotype between a tissue biopsy and and CTCs, which probably represent the leading edge, is that which trumps the other with regard to the therapeutic oncology?
So, sorry, can you repeat your question?
Well, are the two complementary? Do you need both the genomics of a tissue biopsy, which is representative of the solid core of the biopsy, or the edge which is going to bud off and we believe produce the CTCs, which we then analyze to give us a reverse, perhaps genomic or transcriptome of the leading edge, the tumor buds in the tumor?
I think we're just going to get a huge amount of information by doing that. I mean, as the cost of sequencing comes down, and we're no longer talking about even $10,000 genomes, but $1,000 genomes and $100 genomes, we're going to be able to do this, and we're going to get different information from different tumor compartments. And a circulating tumor cell, as you know, is a form of liquid biopsy, but sequencing it is going to give us different information to sequencing a primary tumor, which will contain the stroma, fibrous tissue, blood vessels, and so on. And I think the comparison will be very, very interesting and useful to help delineate therapeutic targets.
Thank you.
I think we'll see markers. I think we'll see certain markers on therapeutic tumor cells that are unique and might help us design specific drugs. Because if you can get rid of circulating tumor cells and a primary metastatic tumor, provided that circulating tumor cells and disseminated tumor cells are probably interchangeable pools, you've probably cured the patient.
Okay, and do you think we should distinguish clusters from single cells?
I think we don't fully understand the role of circulating tumor cell clusters at the moment. It's another really good question. We see circulating tumor cell clusters and we don't fully know what they mean, even their prognostic information. We even sometimes see circulating tumor cells within other tumor cells, and we see strange forms and tumors doing strange things. And to me, this belies our lack of understanding of it.
I entirely agree. And for example, we think of the circulating tumor cells via product of EMT, and they should have lost cadherin, and yet they're adherent to each other. And what determines where they settle, and is it the soil or is it a change in the transcriptome of the circulating tumor cells? These are the next generation of research questions as I see it. Do you agree?
I mean, it is the next generation of research. It's also probably the next generation of clinical trials and marrying up response to those trials, because what we saw when we did our trials with lapatinib was you could destroy a circulating tumor cell pool, but that wouldn't affect the metastatic lesions. They were different biologically, and we were seeing surface marker differences in the circulating tumor cells and the metastatic lesions. And so we were giving lapatinib and we were depleting the HER2 positive circulating tumor cell pool, but if the metastatic lesions themselves weren't HER2 positive, they were staying static and they were growing regardless of the depletion of the circulating tumor cells. And so we know tumors are heterogeneous. So whether you need to treat patients in different compartments is an interesting question as well.
Yes. Nathan, Justin and I can fire off questions at each other for ages. Are there questions from the audience?
Yes, I will read some out, and then what we can do is I suggest that we do an audience question, a Dr. Plowman question, and then interchange. So I believe I know the answer for this one, however, I'll ask it anyway. So Dr. Ungoga asks, with the rise of blood-based DNA testing and protein analysis, how does this fit in with the usefulness of CTC analysis?
Sorry, say that again?
With the rise of blood-based DNA and protein analysis testing, how does this compare to CTC testing?
Shall I go first?
Yeah, you go first.
Cell-free DNA gives you the mean of the mean DNA analysis of the dead tumor cells wherever. CTCs may represent this as I said, leading edge population, the population at the periphery of the tumor, which is going to seed. And so it does give us a difference from cell-free nucleic acids, which, as I say, I think they're the mean of all the tumor cells. Would you agree with it?
Completely. I mean, it's interesting because 5 years ago, you know, cell-free DNA and CTCs were regarded very similarly as liquid biopsies, and cell-free DNA is taken off in that it gives rapid sequencing information. It gives a string of letters, basically, in the same way gene sequencing has taken off more than proteomics. But I think then they are different. Cell-free DNA has not been shown to have any utility in diagnosis. It's not been shown to have prognostic ability, but it does help with prediction and with understanding tumors better. But they are different. I regard them as an overlapping Venn diagram set.
Okay, I think that answers the question well. So going on, so that was the ctDNA as well. So you covered that, and I think that was the difference between the live and then the dead analysis as well. And then in terms of the tumor types, the next question that's came in is looking at CTCs, any top tumor types that you would use this for, or is it literally pan cancer? Or is there any tumor types with which are better for CTCs, that we get better prognostic markers out of it, or what's your thoughts?
Yeah, I mean, can I go first, Nick?
Oh.
I mean, it's licensed in breast, colon, and prostate cancer. If you take breast cancer, for example, if you find a CTC, just one at diagnosis of a primary breast cancer, that confers a worse prognosis, interestingly. But typically they're licensed in metastatic cancer. But what I showed in that dying study, that was all comers with cancer.
So any tumor types that you would focus on, irrelevant of license, we're just in terms of a medical perspective, not looking at different machines now, but where do you feel CTC and assay analysis is most useful in tumor types?
Will I have a go?
Yeah, please.
Okay, I triple negative breast cancer of the subtypes of breast cancer, it looks extremely interesting in, and CMS4 in colon cancer. I think that it looks interesting in that. Those are the subtypes of two common tumors. That is relative to the other subsets on current knowledge, but things may change as we go along.
Right.
But those are both types of tumors, and say triple negative in the CMS or colon cancer, where the EMT is regarded as very important.
And then from a patient perspective, how about the physical getting of biopsies versus a CTC as well? Because then we're talking academically, but the patient is the core of all this, and having biopsies taken is pretty awful. And you know, some locations are harder than others.
Well, you've just used a great pun when you say the patient's the core of all of this, but you know, taking blood from a patient repeatedly is relatively easy. Biopsies are expensive, require specialist labor, need radiology input, painful, risk of bleeding and infection, and we're still very reliant on them. I think in 100 years' time, though, we'll look back and we'll think that we don't really need biopsies, because we're going to use technologies like CTCs to diagnose cancers better, earlier, and treat them better. And a lot of treatments and modern therapies and the way tumors change, you're almost mandated to perform repeat biopsies, but repeat CTC testing, which is not painful and doesn't have all the other issues, is going to be a lot easier. It still remains a true liquid biopsy. Nick?
Yes, I agree with that. I mean, it may be in the future, for example, if you've got a BRAF mutated melanoma, we now treat it with a BRAF mutant inhibitor, raffinib, for example, and then it was shown the most common escape pathway is the MEK pathway, and that there was a level 1 evidence that if you added a MEK inhibitor at the same time as dabrafenib, you not only got an increase in disease-free survival, but overall survival. So we're now mandated to give both these highly expensive drugs from day one. If you can follow the evolution of a tumor over time with serial liquid biopsies, then it may be that you don't need to start the second expensive drug, that you can wait for it to appear and then bring it in. And a similar one with EGFR before we had osimertinib, which covered T90 mutation. We used to use olotinib, and then if you could follow it over time, then you could add the other inhibitor. So I think those are two exemplars of where you can follow the evolution of a tumor's mutations as it evolves very nicely with liquid biopsies. And the liquid biopsies also tend to, as I said before, give you a mean of the tumor overall. CTCs may even give you a finer subset, but a mean rather than the idiosyncrasies of a single site core biopsy of a solid tumor. So those are my thoughts on that.
I agree with that.
Thank you, Nick. Thank you, Justin. Going slightly different question now. So do you think that the measurement of PD-L1 protein on CTCs could allow the use of immunotherapies more effectively? And could they be used earlier in cancer? I'd like Professor Stebing to go first and then Dr. Plowman after.
I'll answer your last question first. So now we're finally seeing some of the adjuvant studies of immunotherapy reporting, which means the use of immunotherapy after surgical resections. And we're seeing some positive data for Tecentriq and lung cancer, and we're going to see more and more adjuvant data, but I think some of those data sets will fail. And what we're understanding is all these molecules, PD-L1 and the next generation immunotherapies, TIGIT or LAG3 or GITR or KIR, they're going to be dynamic molecules that are going to change over time, and we're going to have to measure them in real time. Now, obviously you can sample it. Tumors change all the time, so obviously you can sample a tumor at an infinite number of time points, which is an extraordinary thing to say, but because tumors change over time, you can take blood every single day if you like, or every hour of the day if you like, and understand them. We don't need to know that. But what we do need to know is there's a lot more than response to immunotherapy than just PD-L1. A lot of PD-L1 negative tumors do respond. A lot of PD-L1 positive tumors don't respond. You've got tumor mutation burden as well. And then you're going to have many more things on top of this as well. You've got some immunotherapies licensed in only PD-L1 positivity, and some, such as, say, Keytruda in cervical cancer, licensed in PD-L1 negative patients as well. So it's a dynamic field, and helping to understand responses to immunotherapy, which are actually quite low, better, is going to be very, very important.
And would that be in research or in clinical
Both.
application?
Absolutely both. It's going to start off in research, but by research it really means clinical trials. But it's going to be both.
Okay, then, and then in terms of number of years to start validating that, where do you think that we are on the spectrum of just really looking at the usefulness of it? 2, 3, 6 years out? What's your?
One,
I know it's a difficult.
one or two. I
Okay.
think this is a lot closer than we think. Nick?
Okay.
I'll throw the question back. Do you think we should be looking at the extracellular serum environment for cytokines, chemokines? Yeah.
So I think it's not.
Sorry.
In the same way you can think of a tumor as multiple compartments, the blood is also the same. And you're going to have the cell-free DNA compartment, circulating tumor cells. But you remember I mentioned that circulating tumor cells, or any cells, release little packets of information containing DNA, RNA, protein, and they can actually serve as a barcode to help identify what's going on in different parts of the body. And they can in fact prime metastatic niches as well. We know that from the exosome work performed all over the world.
So a question for Professor Stebing. So are we screening enough in large volumes for CTCs?
That's a good question. I mean, there's a theory that if you're going to look at a primary tumor, say a primary breast cancer or colon cancer, you probably need 30 mils of blood. to increase your probability of finding a CTC. These are very rare, very hard to find cells. But on the other hand, I have a slight problem with such large blood draws. I still think we should stick to the traditional 7.5 mils of blood, and not, it's a bit onerous on patients to have very, very large blood draws, I think. Clinical trials won't allow more than 50 mils at a time, for example.
Okay. And then in terms of the impact of getting the useful information versus negative consequence on the patient, what's the trade-off? Can we move forward without the volume?
I think we can move forward without the volume. I'm pretty reassured if I see 7.5 mils of blood with no circulating tumor cells, and that gives me a lot of reassurance. I don't think I need to repeat that 3 times to be super, super sure.
Okay, Nick? I'm just thinking going down later down the line for looking in where CTC collection happens in the clinics. It's kind of like, because ultimately we're going to get like different CTC collectors with which different companies such as Cell Search and Prostate at Angle, they're going to be in clinics, so we've got to be practical as well as like the academic side of it all, and we need that practical. So I think the volume is an important question. So where would you, if like the analyzers are in multitudes of clinics all around the world, what would you put on the volume to say this is what we need to go for, because it's going to be in so many clinics, because there'll be
I would standardize it 7.5 mils.
Okay, that's interesting.
I would standardize it. Nick might have a different view.
No, I was just musing. You quoted one nice Einstein quotation. Another one, which feeds into not doing a test multiple times, is Einstein saying, "The fool is the man who repeats the experiment 20 times expecting a different result." If you're certain of your data first time round, then it applies to that. So you think that exosomes and non-coding DNA in the serum, things like that, should be included in the next generation of liquid biopsies, do you, Justin?
I do, actually. I really do. But when it comes to exosomes and small, medium, and large and their classification and separation, we're still learning. I mean, some of the data are fascinating about you can transfer exosomes that prime certain areas of the body, like the lung or the liver, transfer it from one mice to another, and the tumor cells spread differently according to the priming of the exosomes, regardless of the surface markers on the tumors. This just indicates that the circulating compartment in cancer is underrated and underestimated.
I agree. Some research I've been recently involved with included, because when you separate off the CTCs as an epiphenomenon, you get peripheral blood mononuclear cells. And it seems to be that their CD45 expression is less in active cancer patients than non-cancer patients or those in remission. And yet, if you co-incubate them with the serum from patients with active cancer, normal patients, healthy patients, peripheral blood mononuclear cells, the serum of cancer patients drags down the CD45 expression on those peripheral blood mononuclears, which I think is it's an interesting observation. Whether it relates to what those peripheral blood mononuclears are going to do in the tumor, or not do, or should do, I think is an interesting research question. Do you think there's mileage in that, researching that?
I do, actually, but there's no one's found a correlation in the way you're asking it right now. Yeah, whether we will find,
except for some of the dendritic cells in the tumor microenvironment seem to have lost CD45 expression, and I mean, are they the derivatives of those cells?
It's you've got the anatomy of the body with the tumor, the draining lymph nodes, the skin, and then you've got dendritic cells passaging between them and tumor cells, and you almost wonder if tumor cells can co-opt the immune system in a way to help it. But
yes,
coming back to the, sorry, Dr. Plowman. So just because the focus on the clinical application today, Paul, got a wild card question coming with regards to the CAR T therapy. Could it be modified to locate CTCs?
I mean, CAR T therapy is a systemic therapy, like a drug. It's going all the way through the body, so it's not going to be selective for any organ or anywhere. Now, the existing CAR T therapies typically target cells expressing CD19 in lymphoma or leukemia or whatever, so they'll deplete all of a patient's B cells, and they'll deplete all the CD19 positive tumor cells and the CD19 positive B cells. And you can live without B cells. You might need IVIG, whatever. But if those CTCs express CD19, and I don't know the answer to that, it'd be well worth doing, then the answer is yes, but they would do. But I don't know quite in a hematologic malignancy necessarily how you'd necessarily define a CTC, because the cells are pretty much floating around the body ordinarily already. It's a circulating tumor, as it were. It's a liquid malignancy. But if they were CD19 positive, it would and should target them. It's a really interesting question.
So we're going to get different questions from the audience, and it may not flow. So if that's okay to just fire the questions at you, is that all right? So do you think there's a role for CTC in addressing the backlog of cancer diagnostics in terms of prostate biopsies because of COVID?
I'll have a go, and Justin can shoot me down. The holy grail of blood testing is the screening test for cancer. Okay, it is unlikely to be based on one analytic, but circulating cancer cells are certainly, in my opinion, highly likely to be one of the analytics we look at in that cancer screening test. The fact that our prostate cancer, which is very early and which is more than 90% curable, it nevertheless is associated with some circulating tumor cells of prostate lineage. The fact that glioma cells, glioma patients have circulating glioma cells, even though gliomas don't metastasize out of the brain, two fascinating observations suggesting that CTCs may well be one of the analytics in a cancer screening blood test. We know what the GRAIL study showed, but I don't think that's going to be the final answer. I think there'll be a lot of analytics, including cell-free DNA, and maybe some of the other intercellular messengers that Justin was talking about and I mentioned. What do you think, Justin?
We're just not there yet. We still need solid tissues to make a diagnosis, I'm afraid. Although you'd think, for example, man with a prostate lesion on imaging, high PSA and circulating tumor cells, you don't need much more than that really, but but you might not get the Gleason score or the number of cores that are positive and so on. Not that I'm sure that's that helpful.
From the RNA in the blood, you could say whether they've got the antigen receptor V7 fusion.
Absolutely.
I actually dodged the question by moving straight to a screening test for cancer. I think Justin can answer the question as posed.
I've got a follow-on question for that, because then as we're focusing on clinical applications, and then in terms of early stage cancers, are we doing these at just, is the future of CTCs in major cancer centers, or is like the way it said there's a few analytics, could they go out to like smaller cancer centers and be used as primary care?
I mean,
how are we going to use all different things?
The Veridex cell search system, which I've got at Imperial, is big, expensive. The consumables are expensive. One good thing about the Prostate angle system is smaller, cheap, much more point of care, easier to use, not much in the way of consumables. So, it depends what you're trying to do and what you're trying to achieve, for example.
Well, if we kind of like look at death as the net result, where can we, in terms of like medicine, where can we have not easy wins, but easier wins? Yeah. So how can we reduce the death rate? Would it be like looking at certain complicated cancers, or can we just start to screen out early prostate cancers to reduce the death rate? I'm just thinking practical terms.
I mean, practically, we probably overtreat cancers. There's many cancers that we diagnose maybe from mammographic screening programs that were never going to cause anyone a problem anyway, and put them on all these treatments and biopsies and yada, yada, yada. I think they'll be helpful, for example, in those patients, weeding out elderly people that never needed their cancer treated in the first place, because they had no CTCs, even though they've got cancers present, for example.
Okay.
Just one simple clinical example, but we're not there yet. Nick, what do you think?
Remind me of the question.
So I was looking at, say there's 100 different cancer types, right? I know there's more. And the net result is to reduce the death rate. So in kind of easier to detect cancers where there's less analytics, you know, where can we use these diagnostic machines and CTC collectors out in, like, say, community, out in smaller point of care centres, rather than just major cancer centres where people have to wait, there's queues, etc., etc. How can we best use this technology to save more lives on a practical level?
Okay, so let's look at the patient who's undergone definitive surgery on ostensibly localized cancer with curative intent. And we recommend for some patients adjuvant chemotherapy afterwards, which is fantastic data for node-positive breast cancer and to start with, and for colon cancer, for example. Now, in breast cancer, we're now doing predictive tests on patients, ER positive patients with 0 to 1 node positive, and they're based on estrogen receptor expression and genes for proliferation, and they're largely based on those two types of genes. And could it be that circulating tumor cells replaced or refined or improved the prediction of those who would be advantaged by some adjuvant chemotherapy? I'm not sure the answer, but that may be where that extra analytic might refine the recommendations for adjuvant chemotherapy. In colon cancer, the patient who has transmural disease but no nodes, say a T3N0 colon cancer, the place for chemotherapy is still very controversial. Adjuvant chemotherapy after resection is still controversial. And there are several tests now which are aimed to refine that. And for example, particularly if it was CMS type 4 with a higher metastatic potential, then it could be that circulating tumor cells could refine the indications or the advantage, could refine the certainty with which you could know that there'll be a statistical increase in long-term survival by giving adjuvant chemotherapy or not. I mean, the advantage would be for those higher risk ones. And so those are two examples where CTCs might provide an extra analytic to help us determine who's a higher risk patient and would benefit from chemo. Justin, your comments on that, please.
I couldn't have put it better, just quite frankly.
Oh, that's
So
no fun.
ultimately, it's about the use of technology to save more lives. And it's, yes, I think that we can do research for the next millennia, but, you know, how can we use what we've got now, you know, to help people with issues now, and then balance that out? Because I don't think we'll ever really fully understand it. So it's, you know, how can we use it?
Well, I mean, I think that the companies that are working in CTCs, I mean, we need a trial. We need a trial where patients are circulating clusters of tumor cells and are otherwise harmonized for the two groups by other risk factors, and look at some extra adjuvant for both groups and see whether the CTCs improve predictions, improve survival, or improve the prediction of advantage. Sorry, that's what I should say.
Makes sense. Question for Professor Stebing. So, do you think the CTCs could be used to detect MRD?
Yeah, I do. I really do. Much more so than bone marrow biopsies. And I think that's really where they come into their own, is maybe after patients, for example, with early breast or early colon cancer or early prostate cancer have been through surgery, chemo, radiation, whatever, if they still have CTCs, then that is a concern, and maybe that would indicate a group of patients that needed more aggressive treatment. but again, it has to be done in the context of a trial.
Okay.
Great.
Another question. So, do you think CTCs will be unseating the current TNM staging system?
I don't think we used it that much anyway, but that would be an interesting addition, wouldn't it?
Okay, fair enough.
Nick, what do you think?
I think my initial answer is no, but they could refine it.
Okay.
Yeah, I agree with that.
So it's back to a lot of Professor Stebing's Venn diagram of information. A lot of these is very likely to just be one data point within cancer, and then the multiple data points with the oncologist making the interpretation from all the data, really. I think that's almost the art of it, isn't it?
Yeah.
And then leading on to the art, in terms of the out in the wider oncology community, when I know you two know this area, but do you see a gap in the knowledge of oncologists out there in to say that, you know, what are CTCs, how to use them, how would you clinically utilize them? I know you two specialize in these areas, but in a gentle way, what about the wider oncology community? How do you think they are in this particular specialty area? In a gentle one.
I think, you know, we make advances in knowledge in small pieces. It's like building a jigsaw, isn't it? And it's a piece of a jigsaw. I think it's an important piece of a jigsaw. Nick?
Yes, exactly. But Nathan's asking us what now, and I think now is we need some trials to show that they improve prognostic certainty. For giving adjuvant or not adjuvant, or indeed for giving one type of therapy than another. And I think that until the bigger centres provide some data to that effect, it won't be disseminated out. You won't get people outside the big centres changing their practice on it. That isn't to say they can't provide very good analytics on changes in the genomic evolution of the tumour over time, or, as we were saying earlier, indicating what the metastatic parts of the tumour, the leading edge cells, for example, are looking like. And that could influence things. For example, the term now in HER2 positive breast cancer is HER2 enriched or HER2 amplified. And Justin in his lecture talked about somebody who had a subset that was HER2 amplified, but not all. And it might be that if circulating tumor cells show HER2 amplification, which isn't seen in the solid tissue biopsy, then that's good enough to give them an advantage with HER2-directed therapy. Do you agree, Justin?
Totally. I think that's right, and I think your enriched term is reflective of the way we're thinking about. I think your term enriched is reflective of the way we're thinking about things differently.
Yes. So there's an example where a genetic change in circulating tumor cells might allow the breast oncologist to bring in HER2-directed therapy, whereas in the face of a solid tissue biopsy with them only 3 months earlier, which didn't show it, and that shows the sort of varying transcriptome.
Are you to being a little biased in terms of the knowledge of the wider community? Because I mean, upon generally speaking, so like once we did market research out to 200 different people, a lot of people didn't know the difference between EMT and non-EMT, or the reverse of that as well. So, and then which different machines could collect EMT cells and non-EMT cells. I'm not quite sure that the wider community in oncology fully understands CTCs at a baseline level yet, let alone the actual advanced area.
I'm not even sure I do. I mean, cancer is incredibly humbling to work with, and I learn every day from it. But I think when it comes to CTCs, I think that studying them is only going to give us more insights about how to target them and how to eradicate them. Nick?
But if there's some level 1 evidence that will have to be produced by big studies that looking at CTCs alters some parameter, whether it's prognostic for the future, whether it's predictive for the benefit of of chemotherapy, whether it's to sort out whether there's a genomic evolution as you go along, whether there's a disparity in HER2 expression in breast cancer, where the circulating tumor cells have it and render the tumor treatable usefully with HER2-directed therapy. Though if a big center, presumably in collaboration with pharma, gets level 1 evidence on that, you'd be surprised how quickly the peripheral oncologists will catch up and become experts. I mean, there's this new drug out 3 weeks ago in the New England Journal for triple-negative breast cancer, and already in a multidisciplinary team meeting last night, there were 2 patients presented who were asked to grant permission for use of it, even though there's an 8-week waiting list for it. So the standard of peripheral oncology is pretty good, but they don't do their background homework and catch up until somebody produces a trial which shows that it's useful, and then they catch up mighty quick. That's my experience.
Understood. And then if you were looking and going out in terms of educating the wider community on the clinical applications of CTCs, you know, let's say that you were a global education company, where would you actually start? You know, what would you say for people to understand, and then just like the top 3-4 points for oncologists out there to start to get to grips with?
They're a much more refined tumor marker than traditional tumor markers. They're prognostic because having a lot means that the lifespan of the patient might really be short, but they're predictive. They can help guide therapies because if you can reduce their numbers, you're on to a winner. Nick?
I have nothing further to add, except they render a lot of research interest in the context of liquid biopsy, which includes the cell-free nucleic acids, exosomes, cytokines, chemokines, and of course CTCs.
And if the wider community understood everything more, then going through the clinical evidence and then getting more lectures out there, do you think that they would use more of the technologies? Would there be a greater uptake?
I think they'd be comfortable adopting it, but you need the kit.
Yeah.
At the end of the day.
Need the kit, and we need the pharma companies, the companies that are engaged in it need to collaborate with big centers to look at trials with specific endpoints, such as, does this predict for adjuvant chemotherapy in Dukes B, that's a T3N0 colon cancer. And if that showed it was positive, it could change practice, and the peripheral oncologists will catch up very quickly.
I agree with that.
And then a couple more final questions. Looking at Professor Stebing, I know that you looked at relapses as well. So is it a good guidance on, could it be monitored to guard against relapse?
Yeah, I mean,
what have you found?
So what we found is really 2 things. Number 1, circulating tumor cells increase before radiologic lesions grow. So they're an earlier indicator of relapse. But also, there's something very interesting going on. There might be this phase of a patient's malignancy in between a primary malignancy and metastatic disease, where they don't have overt metastatic lesions, but their CTCs are going up. And really, my dream trial would be to take those patients with primary cancers that have been treated, increasing CTCs, and randomize them to treatment X or treatment Y to see if you can prevent metastatic disease. Now, if you can do that, you're essentially curing patients who are going to relapse.
Dr. Plowman?
I'm going to avoid the question. I'm going to tell you 2 interesting anecdotes of the phenotype changing as you go on. We had a patient with a neuroendocrine tumor with somatostatins receptor expression, and the primary tumor was very hot on the dotatate scan, which scans for somatostatin receptor expression, and yet the liver mets, which were contemporaneous, none of them out of about 20 had any somatostatin receptor expression. So somewhere between the primary and the development of liver metastases, the somatostatin receptor expression totally disappeared, and we proved it was neuroendocrine liver biopsy. So was it at that leading edge before they budded off? Was it in the serum, or was it the soil of the liver? What changed that phenotype? So that's one thing that makes you think that it's worth exploring the blood environment. The other just anecdotal observation is I do I've done quite a lot of gamma knife work for small brain metastases, and we've got several patients, mainly with breast cancer, who have multiple small brain metastases that we go to town on the gamma knife. We might treat 20 metastases, all small. It's not a technique for large metastases. And we've got several patients who, and I started off very negatively, saying that somebody with 20 metastases has got another 20 incipient behind it, a little lower. But very interesting, these patients have, these selected patients, or a small minority, they've gone 2 years plus before they have another shower. And it shows that something, to my mind, and I think it's contemporaneous, because the second shower all are roughly the same size on MR. And it does suggest that there are times in a tumor's natural history where it showers off some cells with metastatic potential to the brain, and then there's a period where they're not. So they're anecdotes, they're not by no means the majority, but it makes you think about what's triggering the metastatic cells. So those are two anecdotes which I think are food for thought for the audience to take away and cogitate on.
Thank you, Dr. Plowman. So, Professor Stebing, Dr. Plowman, thank you for an exceptional talk. And I think, in summary, the clinical use of CTCs, there's many different uses to explore, and looking at the different early cancers where we can minimize the analytics, where there's only a few, that could be an area where that could be explored more, because ultimately it's about to save more lives. So thank you everyone for attending. So thank you everyone. Professor Stebing,
thanks
pleasure
for having
always.
Pleasure.
And thank you exceptional much to Angle and Prostate, who sponsored the event and who helped to bring everything together today. So thank you to Angle. Thank you everyone. Good night.
Bye.